Welcome to the Sans Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Make sure to like this video and subscribe to our channel. It really helps support the Sans Science Experience. Okay, so let's dive right in. Today, we're unpacking something pretty huge. It's a study involving whole genome sequencing for nearly 500,000 people from the UK Biobank.
That's right, 500,000.
Now, you might be thinking, UK Biobank, haven't we heard about that before? And yes, you definitely have. It's this amazing resource.
It is. But this is different.
Exactly. This isn't just more data. It's like going from a blurry photo to super high definition. Researchers are calling it an unbiased and complete view of the human genome for the first time, really.
And the scale here, it's just, well, it's kind of staggering. Think about it this way. Compared to the older methods, like imputed arrays, this new data set found an 18.8-fold increase in genetic variance. 18.8.
Yeah, okay. That's a lot.
And compared to whole-exome sequencing, or WES, which just looks at the protein coding bits, it's over a 40-fold increase.
40-fold. So we're seeing much more.
We are. We're picking up things that were basically invisible before, especially these rare non-coding variants.
The stuff outside the main genes.
Exactly. And also structural variants, bigger changes like deletions or insertions, stuff WES often misses entirely.
Okay, so this is where it gets really interesting, right? With all this new detail, what kind of insights did it actually unlock? What can we now see that we couldn't before?
Well, it's about precision. Yeah. The study massively improved fine mapping of genetic links to disease.
Fine mapping, like zooming in on the exact cause?
Precisely. Think of it like sharpening the focus. For example, they found this rare frame shift variant in FOXE3.
Okay.
And it was significantly associated with a specific type of cataract called other cataract. Now, this link, it just wasn't seen in the older, less complete UK Biobank data.
So a direct link, previously hidden, now clear because the whole genome approach.
That's it. It reveals connections we just couldn't make before.
Amazing. And does this better view also help with understanding genetic diversity? Because that's always a big topic, isn't it? Making sure research benefits everyone.
Absolutely crucial. And yes, it does. The team did a multi-ancestry meta-analysis.
Yeah.
And they found 126 genetic associations that were actually more significant in non-European ancestries.
Even with smaller sample sizes for those groups in the biobank.
Exactly, despite the smaller numbers. A really clear example is RS334 in the HBB gene.
That's the one linked to sickle cell disease.
That's the one. It's much more common in people of African ancestry, partly because it also offers some protection against malaria.
Right, the protective effect.
So seeing that association stand out so clearly, even in this data set, just hammers home how vital it is to have diverse genetic data. We needed to understand disease risks and, importantly, protective factors across the globe.
It really paints a much more complete picture. So, okay, let's bring it all together. What does this huge new data set mean for the future, for, developing new medicines or improving healthcare?
Well, the paper puts it quite clearly. This resource will facilitate the discovery of diagnostics and therapeutics with higher efficacy and improved safety profile.
So, better drugs, safer drugs.
Yes, and it enables precision medicine strategies like never before. Think about identifying human knockouts.
People who naturally lack a working copy of a certain gene.
Exactly. They're like living, breathing experiments that tell us what happens when a gene isn't active. Incredibly valuable for figuring out if a gene is a good drug target.
Like a natural model system.
Spot on. Plus, they found clinically relevant structural variants that were missed by other methods, like a specific 52 base pair deletion in a gene called CALR.
Okay.
It causes the protein production to stop too early, a stop gain. And it's linked to certain blood cancers, this deletion. It wasn't found in the exome sequencing data, even though it's in an exon, a coding region.
Wow, so WES just missed it.
Missed it completely. This really shows the power of whole genome data for finding these crucial, sometimes tricky, variations that drive disease. It empowers truly tailored treatments.
This deep dive, then, it really highlights the enormous potential here, especially for understanding all that rare non-coding variation.
Which has been relatively understudied, yeah.
It feels like we're just scratching the surface of what whole genome sequencing at this scale can do for medicine, making it more precise, more personal.
It's definitely the direction things are heading, a really exciting time.
Well, we hope you gained some surprising insights today. It's truly remarkable work. Don't forget to like this video and subscribe to our channel. It really helps us keep bringing you these deep dives and supports the Sans Science experience. See you next time.